All right, so I will be talking about genetic isolation by ecological and geographic distance. And I'll be talking about some new statistical methods we have for disentangling those, and a couple of applications to systems in which we think about host-associated differentiation. Okay, when we look across the landscape, we often see that populations differ phenotypically. So here, as an example, we have these red mice and these blue mice. And then sometimes those phenotypic differences are correlated with differences in genetic structure at the population level. But what factors underlie that structure? So given this distribution of phenotypes and genotypes on the landscape, there could be a number of different influential factors, including differences in the habitat that I'm representing here with these different vegetation communities. There's this geographic barrier that is maybe difficult for organisms to disperse across. And then genetic structure can occur just because individuals are far apart in space. So that last one we generally call isolation by distance, and it occurs when individuals closer in space are more related to each other, simply because mating and dispersal often occur locally. So these two blue mice are more likely to mate with one another than they would be to mate with one of these red mice. And we think of isolation by distance as a nearly universally occurring phenomenon across populations. But there can also be isolation by ecology or environment. This is kind of a catch-all set of terms to describe any situation in which individuals in more similar environments are more related to each other, or individuals on the same side of some discrete environmental variable are more related to each other. And that occurs because there's something about the ecology or the environment that affects who mates with who, where offspring are able to disperse to, or the selective environment. So if these blue mice wouldn't be able to survive in the dark gray habitat, or they would not be able to mate with individuals in the dark gray habitat, this can cause these different population structure patterns, even when physical distance is not a factor. But very often in nature, it's not surprising that both ecological distance and geographic distance come into play when we're interested in predicting genetic distance. And they can be really difficult to disentangle. And that's often because different ecological or environmental factors are not uniformly distributed across space. So the geographic barrier between the mice is completely correlated with how far apart they are, and different vegetative communities that these grasshoppers might be adapted to are also not overlapping in space. So if we want to do that disentangling, enter BEDASSLE. This is an R package that implements a statistical model that helps you to understand patterns of relatedness across space. This is really the work of my former postdoc advisor, Gideon Bradford at Michigan State University. I've just helped with really the 2.0 version of BEDASSLE that I'll talk about in a minute. But BEDASSLE came to be in 2013. It's been used in a variety of systems, And the acronym stands for Bayesian estimation of differentiation in alleles by spatial structure and local ecology. So that's very descriptive of what it does. And at the highest level, we're just trying to understand how genetic relatedness is a function of how that relatedness decays with units of geographic distance and units of ecological distance. So why do we need BEDASSLE 2.0, and what are the advantages of it over the original BEDASSLE that's been used in the past? So it's much faster. This is in part because we're now modeling this allelic covariance between individuals using a Wishart distribution. The important thing about that is that calculations still scale with the number of individuals that you're trying to include in your study, but they no longer scale with the number of genetic loci. So it's now more possible and appropriate to use BEDASSLE with genomic scale data. And we use STAN to implement very fast MCMC-based inference methods. And then importantly, for the user, you can now do model comparison, and this is done by cross-validation. So you can ask whether which of these different factors, whether it's isolation by distance, isolation by any number of ecological variables, or both, are necessary to best predict that decay and relatedness across space or other distance. So I'll quickly go through what k-fold cross-validation is, because it is important to understand how the model comparison works. When doing k-fold cross-validation, you divide the loci in your data set into partitions, and you do that randomly. So here I'm doing k-fold cross-validation, or four-fold cross-validation, so I'm dividing my loci into 4 partitions. And I will set 3 of those partitions as training data and 1 as my test data set. But then I'm going to repeat this 4 times such that every locus is included in the test partition at some point. Then we repeat this, shuffling the loci into different partitions several times. And BEDASSLE reports the log likelihood of the test partition data given the parameters that you estimate from the training data sets. And then you compare the distribution of those resulting likelihoods across the replicates in order to choose the best model. And you can compare those distributions using whatever type of statistical method you like to use to compare 2 distributions. So what I'm going to show is basically just using a t-test. So not that complicated. And then once you've chosen your model, you estimate your parameters by running that chosen model on the full data set. So because this is sort of a new implementation of this statistical method, we first wanted to test it using simulated data. So we used SLIM, which allows you to do forward time population genomic simulations. We like to call these SLIMulations. And I here made a simulated population that is under the influence of both isolation by distance and isolation by this environmental variable. So we have individuals that are mating and dispersing locally. But then there's also this discrete barrier that could be anything, but all it does is make it difficult for these simulated organisms to disperse across. And so in this example, I created a really, really strong geographic barrier there. So when we look at relatedness across space, we can compare individuals on the same side of the barrier, that's represented by black dots, on opposite sides of the barrier, that's represented by red dots. You see it's very strong pattern in relatedness. However, there was that local mating and dispersal built into the simulation. So even though its effect is really subtle relative to that of the barrier, we expect and hope that BEDASSLE will be able to reject all models except for the full model. And what we find when we do the cross-validation is very much in line with what we would hope. Here you can see the 4 different models, so a null model, an isolation by distance only model, an isolation by ecology model only, and the full model. And when you look at the predictive accuracy, it's clear that the null model does a very poor job. The isolation by distance only model does better, but not nearly as well as either of the models that include that strong barrier to dispersal. But then if we zoom in on those, it's very clear that the distribution of likelihoods is not overlapping, and that full model is slightly but significantly better in terms of predictive accuracy. And then when we look at that prediction, we have here the 95% confidence intervals around the relatedness between the individuals and the observed relatedness, and it does a pretty good job and picks up that really subtle effect of just geographic distance. So then I wanted to apply this updated version of BEDASSLE to a real-world system. And so the first we'll look at is creosote ploidy levels. This work is really done by Tim O'Connor, postdoc at the University of Chicago. I really just applied his data to the BEDASSLE model, but I think it makes a really good illustrative example. So creosotes are diploid, tetraploid, and hexaploid, depending upon what portion of the geographic range you're in. And creosote is known for having these really specialized herbivore insects, and that's in large part due to their chemical defenses that they have. But many traits important for these herbivores actually differ depending upon the ploidy, including the level of defense chemicals produced and the patterning on the bark that may be, or on the stems that may be important for herbivore cryptic coloration. So we wanted to know how population genomic structure relates both to geographic distances and differences in host ploidy. And Tim and others have looked at this with some creosote specialists, like this Butetics species, that show really strong phenotypic and genotypic divergence on diploid versus polyploid hosts, to the point that I believe they're even sometimes considered different subspecies. However, there are a number of other creosote specialized insects, including this katydid that we'll look at, where you don't really see that strong phenotypic divergence. So we wanted to see if population structure at the genetic level is happening in this species. And so we looked at individuals collected across the range here. They're colored on this map by the ploidy level, and you can see that geographic location and ploidy level are almost entirely dependent on one another. There are just narrow regions where you can find both individuals of 2 different ploidy levels in the same sort of plot. When we look at the observed patterns of relatedness versus geographic distribution, distance, we see that there seems to be very little effect of ploidy in this case. So we have individuals found on the same ploidy level hosts in red, and those on different ploidy level hosts in blue. Doesn't seem to be much effect there. So we want to know, though, if BEDASSLE can actually choose a less parameter-rich model, especially when the geography and the ecology are so tightly linked here. And what we find when we do the cross-validation model comparison is that, this is real data. There's a lot of noise here. And the effects are all very subtle, as you can see in the observed data. But we are actually able to choose the IBD only model. Both the IBD and the full model show higher predictive accuracy than models that don't include isolation by distance, but there isn't actually a benefit to including ploidy model if you're trying to understand the genetic structure for this particular creosote specialized herbivore. So now I will apply the BEDASSLE method for understanding population genetic structure to Phoradendron californicum. I'm going to go into a lot more depth on the ecology and our background knowledge for this system, both because I think it's really interesting and because it is the system that I have worked on empirically. So Phoradendron californicum is desert mistletoe. It is an aerial hemiparasite in the Santalacea of primarily leguminous trees and shrubs in the lowland, desert, grassland, and shrubland of basically the entire Sonoran Desert and a portion of the Mojave Desert. You can see on that map there sort of roughly what the range is. And you may be familiar with it if you've observed it during the winter, that it produces this huge number of really small flowers and red berries that are important food sources for many of the desert birds and pollinators. But there are many factors that may influence desert mistletoe spatial population structure that we might be interested in. So first, of course, there's a pretty broad geographic distribution here, so we would expect to see some isolation by distance. There's a complex biogeographic history of the Sonoran Desert, in particularly of the Baja Peninsula. As I have alluded to already, in that it's a parasite, it also interacts with pollinators. It has dependence upon interactions with other species that might influence its structure. And then, of course, there are climatic or other environmental variables that could be important. So what do we know about what structures Phoradendron californicum genetically at local scales so far? So Phoradendron californicum sits at this web of positive and negative species interactions. The most obvious of those is that it's a parasite, so it has a number of host species that it relies on. We often think of parasites as needing to be adapted to their host species because they spend their entire lives on them. They're completely dependent upon them. In this case, mistletoe is cannot establish without a host individual. So a way that adaptations to host can be maintained is when there is reduced gene flow leading to population structure between populations on different host species. For desert mistletoe, like I said before, it's the Fabaceae, often associated with nitrogen-fixing bacteria, dominant tree and shrub species that mistletoe is primarily parasitizing. So there's a similar ecology to a lot of the hosts. So does host species matter to them? So I asked this by looking at the genetic structure, and I see, looking at a structure plot here, that there is really strong host-associated differentiation. So to familiarize you with a structure plot, along the x-axis, each vertical bar is an individual mistletoe. The colors of those vertical bars represent the estimated ancestry proportion from each of two identified genetic clusters. And I have this black bar here denoting whether individuals came from a cat claw acacia host or a velvet mesquite host. So yes, there's clearly strong differentiation, but this study was based only on a small number of microsatellites and 3 distinct sites in southeastern Arizona. So we do not know whether this is representative of the entire range of mistletoe. But what are the ecological factors that might be causing this reduced gene flow that is allowing for population structure? One could be seed dispersal. Desert mistletoe relies on this uniquely specialized seed disperser, the Phainopepla. Phainopepla reciprocally rely on mistletoe berries for their diet between late fall and spring, and they actually guard territories of mistletoe in the low desert during that time. And they have these gut adaptations that make them particularly effective desert mistletoe dispersers. Unlike many of the other consumers of desert mistletoe, their crop is very unlikely to destroy the seeds during digestion. So the Phainopepla could contribute to population structure through host preferences if the Phainopepla prefer to visit hosts, mistletoes on hosts of one species versus another. That could lead to reduced gene flow. Or if mistletoes on different hosts are fruiting at different times, then Phainopepla would be visiting different hosts at different times and therefore depositing seeds on different hosts at different times. I will just go ahead and spoil this and say that Phainopepla don't care, and there's no difference in fruiting time dependent on host. Pollination could also be an important factor. Mistletoe are dioecious, so pollen and fruit are produced by separate individuals, and desert mistletoe is insect pollinated, so it really relies on this generalist suite of insect pollinators that include flies and solitary bees, other small bees, and honeybees are particularly common pollinators. So pollen flow between mistletoes on different hosts could be constrained, and therefore population structure could be maintained if there are differences in flowering time or in the pollinator community that visits mistletoes on different hosts. So do they flower at different times? Here I looked at the proportion of flowering individuals in the population for mistletoes, again on velvet mesquite and on cat claw acacia. And we see that they do, in fact, flower at different times, with mistletoes on mesquite flowering about a month and a half later. And this should have implications for population structure. It's much more unlikely that a male mistletoe on mesquite is going to be able to deposit pollen on a female mistletoe on acacia than it would be able to on a female mistletoe on mesquite. Do mistletoes on mesquite and acacia attract different pollinators? If different pollinator communities are moving between only mistletoes on different hosts, that should also affect the population structure. Here I'm showing a distance-based redundancy analysis. So this is really similar to a PCA. Each point represents a community of pollinators that visited an individual mistletoe while I observed it for a 20-minute period. And the points are colored based on whether the mistletoes were on cat claw acacia or velvet mesquite. And we can see that there are a few different pollinator groups that do preferentially visit mistletoe on mesquites or the reverse on acacia. So unlike dispersal, pollination may be reinforcing host-associated structure at the local scale in southern Arizona. But what do we know so far at the range-wide scale about population genetic structure or the ecological factors that might be important for it? Just looking at the range of desert mistletoe and of its hosts, we see that those host species ranges are distinct from one another. So there is, correlation just with geographic location and host species identities, but they're also largely overlapping. So this means that it could potentially be difficult to disentangle the effects of different host species and of geography. There is a prior study also by Andres Lara Noriega, who looked at chloroplast haplotype networks across the desert mistletoe range to do a phylogeographic study. And he tested a number of different hypotheses and how what best fit those chloroplast haplotype networks. So he looked at different hypothesized historic geological vicariant event boundaries across the mistletoe range, as well as other environmental factors like vegetation, elevation, and niche centers for the mistletoe. And what he found was that of all of those different hypothesized relationships, the chloroplast network was best explained by this particular set of geological vicariant events, where Cabo is a distinct population than basically the rest of southern Baja, then Baja California, the northern part of the peninsula, versus the rest of the range, basically Sonora and the desert southwestern United States. So then that tells us that potentially host species and historical barriers are really important for desert mistletoe population structure, but we don't know about the role of host ecology at the range-wide scale, or whether those historical barriers correspond to differentiation in genome-wide divergence. So in order to test this, I did a rad capture study and then applied BEDASSLE to the results of that study. So first, some exploratory analysis. Here in the center, I have a PCA of the genetic variation. The coloration of the points is actually the third principal component analysis. But in general, individuals closer represented by points closer together in space are more related to one another than those far apart. And then I've colored the location of the individuals in the study on the map and have done a preliminary splits tree analysis here. So you can see that there's this strong differentiation of individuals at the southernmost end of the range, and then kind of multiple clusters in the rest of the range. So is this indicative of a role of those historical barriers that that previous chloroplast haplotype network study indicated? And then looking at host species across the range, this study looks at far more than just the velvet mesquite and cat claw acacia. But again, we see that the prosopis species, so the mesquites, are really coming out as different, particularly on this second principal component. And this corresponds to some other published and unpublished prior work of mine that suggests that it's really mesquite that is quite different in terms of a host for a desert mistletoe than all of the other hosts. So there's something about living on mesquite that is much different than living on either acacia or palo verde or ironwood, etc. So does this indicate a role of host species in determining spatial patterns of relatedness? And how do both the historical barriers that may be present and host species distributions relate to the role simply of isolation by distance? Because you can see some geographic patterns, obviously, in both of these different factors. So in order to test this, we looked at 8 different models to compare with BEDASSLE 2.0. There's the null model, one that includes only geographic distance. So here we're looking at the effect size of distance that is in the unit of 1,000 kilometers. So that gives you a sense of what these effect sizes that will come out will look like. Then we looked at a model that includes only historical barriers. So this is coded simply as if you're on the same side of one of these barriers, then you have a distance of 0 for this variable, and if you're on different sides, you have a distance of 1. So this is a discrete ecological variable. Similar for hosts, and so I'm just grouping prosopis species together versus everything else. So if you're in the same host group, you have a distance of 0 in the host variable, and if you're on a different host group, you have an ecological distance of 1 there. And then we look at all combinations of those different factors up until the full model that includes all 3 factors. When we do a cross-validation model comparison to look at the predictive accuracy of these different models, there's actually a slight but significant increased predictive accuracy of choosing the full model with all 3 factors. However, you can clearly see that geographic distance and host are contributing more than those major that should be breaks from historical geological barriers. So there's very little benefit of including the historical barriers beyond just looking at geographic distance and host species, but it is significant. And then if we look at the model fits here, the colors represent whether you're in the same host group or the same side of a barrier, and plotting relatedness across geographic distance here. And then when we really zoom in on the right-hand side of the screen here, you can see that there is a clear effect of different host group. So you're more related to each other if you come from the same host group. But that effect of historical barriers is really small, despite it having been the best fitting factor in that previous chloroplast haplotype network study. But of course, that study doesn't take into account the effect of just geographic distance itself. Then when we just look at those effect sizes, again, you can see that geographic distance and host species group have a larger effect than the small but significant historical barrier effect. In the future, what I am really looking forward to doing is understanding how the ecological factors that we've determined to be important at the small scale are differing across latitude and or on opposite sides of historical barriers. So you may have noticed that in that really divergent part of most southernmost part of the range, there was actually less differentiation between individuals on mesquite versus other hosts, as opposed to the really strong differentiation in the northern part of the range. So I would like to know if there's less phenological or pollinator community disparity across hosts when you go to lower latitudes. So if you have questions about using BEDASSLE in your own work, please contact myself or Gideon at these addresses. I think you can see that there's a lot of different applications for it. If you're interested in many different types of factors and how relatedness at the population scale might correspond to them. And then quickly, I just want to bring up what I am working on these days. I am now the project manager for the biorepository arm of the National Ecological Observatory Network. So the natural history collections at Arizona State University now house all of the physical samples being collected by NEON. NEON is this continental scale tool for monitoring and forecasting primarily ecological change. And they're collecting over 170 data products that are freely and openly available on their data portal. And the project is intended to run for 30 years, starting last year in 2019. But at ASU, we have all of the physical samples. So these are taxonomically diverse in their potential uses, from genomic samples to environmental samples. So they're really meant to be useful for evolutionary biologists, ecosystem scientists, as well as ecologists. And so please contact us if you're interested in doing projects with these types of samples. I am really interested in the increasing ways that we can use natural history collections to answer exciting questions in both ecology and evolutionary biology. So with that, I want to thank my sources of funding and acknowledge especially Tim O'Connor and Gideon Bradford for being really active collaborators in this work, as well as my primary advisors in grad school when I did much of this work, Judy Bronstein and Noah Whiteman, as well as a number of other collaborators. And